Good morning. It's a pleasure for me to present my work about label-free biosensor for human cell monitoring and the supervision of Prof. Hassan Zairi. My work is beginning with the definition of biosensor and why we use uh, this element. After we will discuss the difference between label-based and label-free biosensor. Cellular, after we will discuss about the cellular interaction uh, and requirements for biological detections. We will focus a little uh, about graphene layer. Mhm. After I will present my prototype and a part of result. Not all of my thesis work, it's a part of my work. What biosensor? Biosensor is an element that detects and measures the presence of specific biological molecules or substance, such as uh, cellu- uh, cellular cells, virus, enzymes. It's a combination between biological sensing and element biological sensing that I have cited, cells or virus or and so on, and the transducer, that is the microchip or the the electrode, the two types. Why we use the biosensor? Because the conventional technique is very limited, expensive, and time consuming. It's the laboratory bound, demands expertise to study the behavior of cells and needs pre-treatment of the samples. Uh, uh, it make it it's making it a laborious task. The biosensor advantages is uh, low manufacturing cost, fast response in time, portability, because the biosensor are a small chip, like a microfluidic, like a microfluidic chip or electrodes in a size of a micrometer. Uh, and biosensor also are specificity and sensitivity, because we use different material such as graphene and gold, and these material are known with their biocompatibility, that not affect the cell. Uh, it furnish continuous real-time signals and provide toxicity level. In this step, we will see the difference between label based and label free, and which is good for continue the work. The label based technique is it's a technique based on the labeling, such as fluorescence, and these techniques is widely used, but the tags like fluorescence may interfere with the function of cells, uh, and it's label based. It's costly. Right. So we will not pursue our work with label based technique. I have pursue the work with the label free technique. The label free technique does not need labeling to test the behavior of cells. Um, it's the advantages in the second figure picture. We can see the behavior of electromagnetic waves, electromagnetic wave um, that will interact with cells and give us the information about the sample. It's very sensitive and low cost. The cellular interaction in study in 19 Haber study the evaluation of the resistivity of blood sample at low and high frequency, and the result is the different response in low frequency and high frequency, that the cells don't have the same permittivity in low frequency and high frequency. Here, picture present the interaction of electromagnetic wave with cells. We can constitute at megahertz frequency the interaction of electromagnetic wave is with the membrane of cell. When we work in megahertz frequency, we will have information about just the membrane of cells that indicate the dimension. Like when we go forward with the frequency in the higher frequency at megahertz, at the gigahertz frequency and terahertz frequency, the electromagnetic wave interact with the host medium and the intracellular compound. The requirements for biological detection. For biological detection, we need a low-cost prototype, label-free, no invasive, very sensitive, and quantitative. That's sensitive for low concentration of cells, like 1 or 2 cells. I have focused in the interdigitated capacitor because this structure can give us very high electromagnetic wave in area of detection between the interdigitated finger. Also, I have added the graphene in my work to augment the sensitivity of prototype. I have simulated this prototype. It's a combination between microfluidic channel and graphene, gold, quartz substrate, and PDMS in the top of substrate detection. This combination provides many advantages, such as improving the selectivity and the sensitivity, achieving a chemical and thermal stability, decreasing the size of device, and improve the cell of this and improve the biological reactivity. Here we find the result. The important thing in graphene is in graphene we don't need to change the material of our prototype is just a parametric study of chemical potential, temperature, and relaxation time. In these results, I have chosen the chemical potential is 0.02. Uh, the temperature electron volt, the temperature is 300 Kelvin, and the relaxation time is 0.5 uh, picosecond, and it gave us the a prototype and adaptive prototype in frequency. I have after tested the medium agar solution that is the cell culture. The agar, when we put the cell in this solution, the cell keep their dimension and their first properties. And after I have simulate a kind of skin cells, I have implant the permittivity, conductivity, and the losses from previous article that they use, that they have uh, that they realize the result in the real. I have take the conductivity and the other parameters. I put it in my prototype, and I have tested it. It shows that our prototype is sensitive for a low concentration of cells, like 2 for the a little, very small cells concentration. Thank you.